slide?
Yes, we can see the slides.
Excellent. Thank you. The title of my today's talk is Modulation of Gamma-secretase for Alzheimer's Intervention. Thanks for Sonia's introduction. I'm going to get into the talk. So I'm going to give you the overview and the research goals. Alzheimer's disease, I'm going to give you briefly the background and the common risk factors, including age, family history or genetics, and sex, a beta hypothesis, also introduced and described by Sonia, focusing on genetics and biochemistry analysis over the past many years. I'm going to talk about President-1 and gamma-secretase, focusing on mechanism and pharmacological intervention. Regarding that, I'm going to talk about the gamma-secretase inhibitors and gamma-secretase modulators that we are starting. In conclusion, I'm going to talk about the GSMs and also the hypothesis, and also going to talk about the future directions. So histologically, Alzheimer's disease has been studied for many years. The Alzheimer's disease is first reported over a century ago, and then it's reported in meetings and the reports for A-beta and tau. And then until 84 and 85, identified for the small molecule A-beta. 87, APP is identified. And then first FAD mutation subsequently identified in London mutation. And then ApoE risk allele is reported. And also President-1 and 2 reported in 1995. Based on that, the genetics and biochemistry, it formed the A-beta hypothesis. And then 2005 till now, linkage analysis and GWAS, exosome genome sequencing, identified new variants in addition to FAD mutations, including ubiquitin-1, ataxin-1, CD33, and TREM2, highlighting the neurons, astrocytes, microglia. In combination, they affect the beta hypothesis, providing our understanding and pharmacological regulation for reducing the beta plaque for treatment. Looking at the Alzheimer's common risk factors, the first is age, and second is genetics. The APP President-1, 2, that Sonia has described, these mutations are causative, and ApoE is increasing the AD risk, and other risk factors can modify the disease risk, including life exposure, stroke, hypertension, and lifestyle, etc. Looking at the genetics, the mutations in APP President-1, many mutations has been identified, and biochemistry analysis showed the increase of A-beta 42 to 40 ratios is a primary loss of gamma-secretase processability of this non-A-beta species, for example, A-beta 42, 43, etc. And ApoE risk allele also cause AD by increasing A-beta neural deposition. Looking at the discovery of genetics, this FA mutations is reported in President-1 and President-2. And biochemistry analysis showed increased A-beta 42, 43 in brains of mice expressing mutant President-1. And FAD related President-1 variants elevates 42 to 40 ratio in vitro and in vivo. And the mutant President-1 of AD increase A-beta production in translated cells and transgenic mice. Overall, the genetic analysis identified over 300 FAD President-1 mutations, which showed this increased ratio primary loss of function in processing of this longer A-beta species. This shows the structural enzymology of gamma-secretase in its active cleavage site and also the complex formed by President-1, N2, nicastrin, and APH1. This is a beta hypothesis that Ksenia also described. It's a missense mutation in presentin 1 to APP causing a beta 42 production and accumulation, and non-calcium dysfunction, microglia astrocyte activation, synaptic dysfunction, oxidative stress, neurodegeneration, and this dementia. So clinically, this shows a beta hypothesis. It can define the disease stage and the biomarkers. And recently, for the brain biomarkers by PET and functional MRI in the blood and CSF, the changes of A beta 42 and phosphorylated tau has been a recent focus in the field. Based on that, we can, this A beta hypothesis have advanced AD prevention and treatment. Looking at here, the A beta change is one of the early in AD progression. It leads to the synaptic dysfunction, and then tau, neuronal injury, and brain structure change. Here, it is early change on the mild cognitive impairment, and then followed by ultimately dementia. Also focusing on this, it also helped us to design our trial in prevention or the treatment. This shows the A beta in relationship to the tau and the inflammation. This shows amyloid genesis can be caused by the overproduction of A beta by APP overexpression, and beta or gamma-secretase abnormality, and also the influenced by A beta clearance or degradation, primarily by astrocyte or microglia. And also because of its aging-related disorder, by looking at PET probes, in addition to A beta, using Pittsburgh compound B and others, it's very useful to design probes for neuroinflammation or aging biomarkers. For example, in addition to TSPO, we are interested to look into the RIPK1 possibility and a certain one for AD-related changes. For PS1 gamma-secretase, here shows the transmembrane domains and the luminal cytosol in this presentin 1 structures and F80 mutations identified. One of the locations is hydrophilic domain HL1 here. It connects exon 4 and 5, which is modulatory key sites for gamma-secretase activity. And here is endoproteolytic cleavage, because this whole presentin 1 does not have active activity. It will undergo endoproteolytic cleavage to form presentin 1 NTF and CTFs to function as active gamma-secretase. The first study and reports after I joined MGH at Rudy's lab is a publication following up Nature reports, APP binds DR6 in relationship to SCP beta. We showed F80 mutations in presentin 1 using a mechanism other than affecting SCP beta change. As you can see here, this is anti-vector, and we transiently expressed different PS1 wild type and F80 mutations. It all increase 40-to-40 ratio. This is earlier this year that Dr. Liu Lei in Dr. Fanny Soko's group reported this study. He mutated up to 17 F80 mutations in cell-based model and analyzed A-beta different species using super-sensitive ELISA kit, homemade. As you can see here clearly compared to the wild-type PRISM1, all these F80 mutations significantly increased 40-40 ratio. And one of the severe variant is 116 R here. In addition to all these previous findings, PRISM1 mouse models has been reported. Here shows you a finding that PRISM1 knock-in mice reveal loss of function mechanism for F8D and a loss of A-beta 43 production caused by PRISM1, suggesting this seems very complex phenotype. And then F80 mutations in PRISM1 generate amylogenic A-beta peptide seeds by Dr. Distroper's group. And then a paper using knock-in rats with PRISM1 homozygous shows these rats are viable, not like mice, they are lethal. We show selective gamma-secretase activity with high A-beta 42 species. So in addition to these genetic findings of PRISM1 gamma-secretase, gamma-secretase inhibitors and modulators has been discovered. This is an earlier finding that identify the active cleavage sites within PRISM1 by Dr. Yuming Li and his colleagues. And GSI is one of the first waves in Alzheimer's A-beta hypothesis pharmacological into clinical trial. Unfortunately, it failed. It shows a phase 3 trial of semaglutide, a GSI for treating Alzheimer's disease, does not show improved cognition behavior. So instead of inhibiting all A-beta species, targeting A-beta 42, which is for early intervention that Sonia showed before the pathology of AD or treatment of AD can be a strategy. Based on the genetic findings and A-beta hypothesis, in addition to gamma-secretase inhibitor, there's also BACE1 inhibitors. Unfortunately, they all failed due to safety issues. Instead, GSMs has been discovered by our group and also in the other groups in the field. For example, we developed a gamma-secretase that can be used as an imaging probe for gamma-secretase. This molecule called 15606, and our 776890 is a trial candidate for AD. There are other strategies, including antibodies and also neural stimulation, that has been under trial, suggesting the efficacy and the safety will be the focus of these trials. So gamma-secretase, what are these? It's a group of small molecules which can access PRISM1 and allosterically modulate gamma-secretase processing of APP and thus ameliorate AD-related loss of gamma-secretase processing and preferentially lower A-beta 42 or longer A-beta species. Our GSM study has focused on studying GSM for potency, safety, efficacy, and also mechanisms. We use different methods, including genetics, medicinal chemistry, cell molecular biology, structural biology, radiochemistry, molecular imaging, etc. This slide shows the A-beta cleavage and a gamma-secretase modulator as a model. As you can see here, A-beta species is cleaved every 3 amino acids, and gamma-secretase is here. It can access and bind GSM with gamma-secretase. And the effect is reducing 42, increasing smaller length of A-beta species, for example, A-beta 38, 37. And here shows GSI gamma-secretase inhibitors in comparison to GSM gamma-secretase modulators. Our GSM has been developed in collaboration with UCSD by Dr. Rudy Tanzi and Dr. Steve Wagner's group over the past nearly 20 years. And we have three classes of main class of GSMs. Here is amylzino group, or called aqueous solubility GSM, AGSM, this compound 4, and this is a second main class. GSM 36 is a key lead compound. And our recent pyridine class GSM is our most newest class of GSMs. As you can see here, the improvement of our GSMs have been aqueous solubility and also the potency of reducing A-beta 42. Here, the slide shows the first generation GSM by acute 3-day treatment in APP Swedish TG2576 animals. Here shows the different dose. Here shows the comparison to the vehicle. This is efficacy in plasma and efficacy in brain. And this is a chronic treatment in APP Swedish TG2576 animals. It showed good safety and also efficacy in reducing plaque and A-beta levels. Importantly, our GSM do not affect notch processing because gamma-secretase, as Dr. Oksana's group showed also with Dr. Sokol, there are many hundreds gamma-secretase substrates. One of the key substrates is notch. Gamma-secretase inhibitors failed in clinical trial. A major reason is safety from notch inhibition. Our GSM do not inhibit gamma-secretase, as you can see here. It doesn't inhibit the APP processing. We used all possible available AD cell models, and also in addition to animal models. So this is our results showing 3D AD human neuroculture model developed by Dr. Duyan Kim, Dr. Rudy Tanzi. Dr. Sehong Choi reported with colleagues that in comparison with DMSO, beta-secretase and gamma-secretase inhibit and reduce plaque formed here, and our GSM also reduced A-beta deposition in this cell model. Differently to BACE1 inhibition and gamma-secretase inhibition, which is compound E here, our GSM41, this is a molecule from the second class GSM. It increased A-beta 38 levels, as you can see here. While increasing this small A-beta species, there's reports that shorter A-beta peptides attenuate A-beta 42 toxicity in vivo using fly models. This is a proof of concept. We can increasing this is A-beta 38 or A-beta 37 as an effect of our GSM with safety profile. In collaboration with Dr. Oksana, we showed the Princeton 1 confirmation in relationship to GSM treatment using threat analysis in cells. As you can see here, GSM 36, that is a key lead compound from the second class of GSMs. It reduced threat efficiency when it reduced the NTF and CTF proximity. And this is published And we show this soluble GSM attenuate AD pathology and induce conformational change in PRISM1, led by Dr. Riven and also Joseph Ward. This is a model published 2015 showing the pharmacological modulation of GSM in this, for example, a hydrophilic loop 1 region of PRISM1, which affects the catalytic, the key region within PS1. When it is modulation activity is led by GSMs, it leads to the reduction of A-beta-42 production without affecting Notch and other gamma secretase substrates. So gamma secretase is an example providing that PRISM activity can be pharmacological modulated. Very interestingly, Dr. Liu's group from Dr. Soko's group showed genetically gamma secretase can be also modulated, considering the Icelander mutation in APP, which can be protective in Alzheimer's disease in human. Here shows a cell-based model that mutation in 106 and 113 in PRISM1. It showed a protective effect in with increasing shorter A-beta species, shorter A-beta species that relatively improved compared to wild type, but with sub-responsiveness to GSMs, as you can see here. Usually for the wild type, all the GSMs are very active. It shows a robust reduction of different GSMs. But with all these mutations, 106 and 113, in screening assay, these GSMs lost at least partially to some GSMs, suggesting these sites can be a positive binding sites, allowing the activity of GSM to have their activity. So with all these evidence, we are working toward a clinical trial potentially for GSMs. And this is 15606, our original molecule for the rhodazine class GSM. It shows a perfect example for reducing A-beta-42 preferentially here, and then reducing A-beta-40, at the same time increasing A-beta-38. And also it showed drug-like abilities. It can be a drug. Using this molecule, we are thinking, where can the PRISM1 or gamma secretase that allowing GSM binding in the brain or in the body? And what our discovery is in collaboration with Dr. Changing Wang at our Martino Center at MGH. The approach is straightforward. We took the GSM, we radiolabel, and when we perform in vitro pharmacology analysis. We evaluate radiolabeled compounds in rodents. We evaluate brain uptake, specific binding, and dosimetry in non-human primates. And we have the ultimate goal to translate into human imaging. And we recently published our finding. So our study describes the development of our radiolabeled 15606 to review gamma secretase in rodents, including 5XAD animals and non-human primates. And then we are working hard for translating into humans. Briefly, this is our specific results. On the left, you can see this is our radiolabeling methods. After radiolabeling, we want to make sure we can self-block our compounds, which is shown here. We characterize AD transgenic 5XAD transgenic mice compared to wild-type mice. These 5XAD mice overexpress APP and PRISM1. So as expected, we showed upregulation of radio signal, as you can see here. The advantage of PET imaging in combination with MRI is we can see the different brain regions in live animals for PRISM1 and γ-secretase, as you can see here. So generally speaking, PRISM1 γ-secretase is a conserved protein that is expressed at different brain regions. But at certain brain areas, it showed relatively higher expression in rodents and non-human primates, including anterior cingulate cortex and midbrain. So in addition to our radio-labeled in PRISM1 γ-secretase, there are other previous studies suggesting radio-labeled compounds can be used in AD etiology. They are suggesting a research that where the γ-secretase can be detected, it can be a signal analog, suggesting for soluble A-beta species. Notably, currently clinical used Pittsburgh compound B for A-beta deposition. They are usually for late stage A-beta deposition, but not for the soluble A-beta. And the soluble A-beta is, as far as I know, there's no PET imaging detecting soluble A-beta in clinic. And in preclinical, it's also been very challenging. So our γ-secretase can be used as an analog signal for soluble A-beta species, allowing for early detection. So PET probing γ-secretase and PRISM1 in clinical have certain advantages. First is there is a clinical needs, but 1% of FAD PRISM1 mutations in the AD, all cases, it's about 57,000 Americans from Alzheimer's Association estimate. So it can provide insight of AD. Our PET tracer may be used to study PRISM1 distribution in human AD brains. It has to be further developed and characterized. Shoot A-beta hypothesis. So with PRISM1 PET imaging, it can be providing opportunity allowing to early detection of this A-beta species as an analog signal. Once developed and utilized, it can be stratified patients and help evaluate drug development and treatment, including our GSMs. So hopefully using our PET probe, we can have early detection of AD progression. It can work with other biomarkers to better understand AD. TSPO and tau and Pittsburgh compound B, all these available AD PET tracers. We plan to use our GSM-based PET tracer to further understand AD. Now we can use our GSM PET to advance AD therapeutics based on GSM. So this is our clinical preparing molecule, GSM 76890, which we recently published it potently reduce A-beta-42 in cells. As you can see here, the IC50 is close to 4 nanomolar. And acute treatment with a single dose of GSM, it can reduce A-beta-42 levels. When we treat with chronic treatment, starting 6 months old for 3 months, we label it as here, it can reduce the A-beta burden detected by 3D6 antibody. It can reduce the microglia reactivity by EBAR1, the signal for neural inflammation. So it displayed good drug-like properties, and IND application is in process with a plan for phase 1A trial, end of this year, early next year. So as a quick summary for our GSM, it provides an ideal candidate, what I think is for testing A-beta hypothesis. It's based on the A-beta gamma-secretase, the loss of function processing longer A-beta species. So we have cell-based studies that are different from GSI. We also performed animal-based studies. We are working on different areas. In collaboration with Dr. Wang, we are working on molecular imaging for GSM-based PRISM1 and gamma-secretase. Working with Dr. Steve Gumpertz, we are working on hippocampal electrophysiology for the neuronal activity. So with all these preclinical cell-based and animal studies, we hope to achieve good safety and efficacy using our GSMs. So there are these different areas that I described in previous slide. So using GSM, we can support a potential trial. We can also further study PRISM gamma-secretase pathophysiology. We can link of our molecular imaging of PRISM1 and gamma-secretase with other AD biomarkers. Because AD, considering its longitudinal changes, we can use this gamma-secretase probe in combination with SIRT1 and RIPK1 that we are currently working on. So while we are waiting for Sonia's MB02 and our GSM, so what can we do? There are other emerging studies focusing on the lifestyle intervention in AD. I have 2 or 3 slides in here. Here shows AD risk can be modified. These are human cohort studies suggesting exercise, traumatic brain injury, blood pressure, smoking, education, air pollution. All these can be modifying risk factors of AD, and with different lifespan, there are different factors identified for population attributable fractions. Here, starting young, there are factors less education can increase AD related to AD risk, hearing loss, TBI, hypertension, etc. Potentially hypertension with calcium dysfunction. And smoking, depression, social isolation. These are potentially modifiable, occupying 40%. There are about 60% unknown risks. So focusing on A-beta pathology subsequently to tau and neuroinflammation, it provides opportunity for us to understand AD pathogenesis, diagnosis, prognosis, and drug development, and we have a hope to prevent and cure Alzheimer's. All this lifestyle or whole body intervention, and also different mechanisms like calcium dysfunction, homeostasis, and A-beta-42 reducing GSM. I did not show you, but we are also interested in other mechanisms, including curcumin analogs and HL-XL-based natural compounds they can modulate immunity themselves. So they can constitute as polytherapies for Alzheimer's disease. As a conclusion, Alzheimer's disease, I gave you the background, and I also described the common risk factors, the A-beta hypothesis. Using A-beta hypothesis, I described the A-beta 42 reducing GSM, focusing on mechanism intervention. So our GSM attenuate neuropathology of AD. It can reduce longer A-beta species, particularly 42. It can reduce A-beta deposition and tau phosphorylation in our 3D human neural culture. It can correct the neuroinflammation in AD. It can improve the cognition in preclinical models. And we are preparing and getting ready for a human trial. And future directions will be advancing our drug development and also probe development to have a better and a complete understanding with other processes in AD. So this is cartoon from this year for oncology. This is next generation from this on the right of cancer, but I use for Alzheimer's disease. It's for translational medicine of proteogenomics of AD. One is looking out in a brighter horizon where disease practice is achieved by multi-omics studies, genomics, proteomics, pharmacometabolic omics, and others, which may work in combination and a synergy from preclinical studies into clinical trials, and ultimately advance patient care of AD. On the left, we start with AD subjects and control. We identify mutations and also genetic variants. We translate using cell and animal-based models with multidisciplinary approaches, and hopefully we can have prevention or cure for AD. With that, I want to acknowledge our institution of Mass General and my team, Dr. Rudy Tanzi, Stephen Choi, Dr. Tanzi, and Dr. Oksana, Steve Wagner, reported GSMs, Dr. Channing Wang, Dr. Xu, worked on GSM molecular imaging at Martino Center. I recently have group members, Dan, Dr. Mandel, and Ashley. I want to thank my collaborators. And this is our unit after lunch possible a couple of years ago. This is our GSM study for this group, wonderful group. And also want to thank our funding, Cure Alzheimer's Fund, EDF, and NIH. This shows my Alzheimer's journey, research journey, starting with Dr. Alistair Saunders at Drexel University many years ago. I really appreciate the mentorship and all the leadership and the support for over the many years. And also GSM also started with a Spark funding for studying the sleep-related electrophysiology change. Finally, I want to thank the committee again and for our audience and participation. Thank you, everyone. I'll stop here.